Okay, so let's say that we've got a plant that we've basically cleaned up. I showed you another video about that. And this plant is mostly clean of associated species. We got lots of fresh green parts of this plant. This is its own piece of a plant, and then this is another full segment of the plant. And keeping intact parts of the plant are a good way to ensure that at least the tips of these plants are going to be a little more intact. The plant can divert resources or sort of sacrifice older tissues to save the newer tissues, and so the newer tissues, what we'll probably use for DNA work. Now, this is still a lot of material. This is a good amount for making a voucher specimen, which is a valuable part of the process, where we can basically dry one and use it as a permanent record of wherever the plant was found. So if we go through the process of identifying a plant, we'll have a record that that plant from that place was identified in a certain way. But realistically, just to do the DNA work, a single leaf would probably work for us here. We just want to kind of stack the deck in our favor to have as much material as we can to make this work moving forward. So this little bud down here would be probably where I would target younger tissues, less contamination, less denser cells. That's a good place to get DNA from. All right, so let's say we have this all cleaned up and we want to pack and ship it in a like a Ziploc container. I got a Ziploc back there, and really there doesn't have to be much to it. You can just put it in a Ziploc about like that. The amount of wet is actually pretty good right here. So we don't have any standing water. A little bit of standing water in the corner there. I might pour that out. The standing water is a location where rotting can happen. So it creates zones where other organisms can thrive and start picking apart the plant that you're trying to send. Also can create kind of an anoxic, oxygen-free environment that also can contribute to some rotting. And another thing you could do in this process is to put like a damp paper towel. So let me grab one. And people think on first pass with water plants, they might have an impression that they should send it with a lot of water. And certainly these plants thrive in water in their own natural environment, but during the sending in the mail, that's an environment that we want to really minimize the opportunity for rotting. So I've got just a simple paper towel here, and I'm really just wringing it out a whole bunch. So it's not very wet. You can see it doesn't drip as I hold it up. It's just moist. And this, combined with the airtight Ziploc container, is going to give us basically 100% humidity. And so 100% air humidity isn't so different from actually being in the water as far as letting these plants stay hydrated. So the paper towel in here, this might actually do us the favor of soaking up some of that extra standing water. And I'm going to do one more thing, and I'm going to actually blow in some air, kind of like if you ever buy a fish from the pet store. They put air in the bag that's airtight there, and that also can help to kind of space things out and keep the rotting isolated if any of it happens. It's going to be hard for me to show that on videos, so just imagine that I've blown some air in here and then sealed it up that way. And then, of course, as you send these, include some kind of identifier. A Sharpie on a Ziploc is pretty good. Otherwise, you could do a pencil-written piece of paper inside of the bag, or any combination there. As long as I can identify, distinguish your different samples as they arrive, that should be good enough. Thanks.